Hello, good afternoon. Thank you for being on time. We're going to go ahead and get started. What we will be doing is take up the review package that I post online for test one. Of course, test number one is going to be happening on Thursday. And there will be no class on Thursday. You just have to do the test. So let's get started. First question about the Beetroot Lab. What is the control of the experiment? Right? The control of the experiment is the room temperature. The room temperature test tube. Okay, that's the one that has not been modified. You did not heat it up. You did not cool it down. It's just the way it was. And it's important to include a control because we want to have a reference, right? So provide a reference to compare with. Okay. I will of course post this online afterwards. So if you miss anything, you can download the PDF after. What is the dependent and independent variable? You should know by now for the Beetroot experiment, the dependent variable is the color intensity, which was measured qualitatively using sight as well as quantitatively using the colorimeter. Right? So it's color intensity, or you can say absorbance, which would be an indication of how intense the color is. As for independent variable, right, that would be the temperature. What variables are standardized? So these are things that are kept the same, right? So we have the size of the cut beetroot. Okay, you want it to be similar size with same surface area, right? So that they're not going to be leaking more. Like if it's one is bigger, the other is smaller, and obviously the bigger one will potentially leak more color, right? So you don't want that to be interfering with your result. Time.
Excuse me, sir.
Yep.
Oh, it's the size of the what?
Of the cut beetroot. Yeah.
Thank you, sir.
Time spent in the water bath, right? The 20 minutes, right? If you put it, if you leave it in the water bath for too long, then you might have an overdevelopment of color. Everything eventually leaks out if you give it enough time, right? And if you leave it in for too short, you prevent it from reaching the temperature, and you're not going to have sufficient color development, right? So you just need to leave it in for the right amount of time. Volume of water in test tube. Right? You want each test tube to have the same level of water. If you add too much water, then you're gonna dilute the color. If you don't have enough, then you might make the color more intense, right? So these are some standardized variables. Okay. Why does a frozen beetroot have similar color intensity as 80 degrees? Right? So you should know by now, at 80 degrees, the cell membrane is pretty much destroyed and all the color leaked out. And this question is asking, why would a frozen beetroot have all the colors leaked out. Like, I mean, you didn't heat it up or anything, right? So in class, we talked about how freezing a cell is going to damage the cell membrane. So ice crystals formed during freezing damage the cell membrane. So your cell membrane is basically full of holes, right? And when you thaw it and put it in a test tube, all the colors just leaked out. Right? You don't even have to heat it up to damage the cell membrane. You don't even have to heat it up to make the cell membrane leaky. It's already leaky because the ice crystals, it's like poking holes in it, right? Okay, for those of you just joining us, I am taking up the review sheet. The stuff that you missed, I will post online after, so you can copy those down later. What do you expect the absorbance to be if we include temperatures higher than 80 degrees? Right, so like the graph, some of you probably didn't like read the instruction carefully. I actually posted a video that shows you how to do the graph, right? I don't think all of you pay attention to that, but you should be using a line graph, and the line graph looks something like this. This is the absorbance, and this is the temperature, right? So during this phase, basically, you have the increased kinetic energy in phospholipid. Okay, so the phospholipids are like they can gently move past each other, right? But when you heat them up, they like move faster, and then that creates more gap, and color starts to leak out. And then what happens is during this phase, right, protein starts to denature. Membrane protein starts to denature, and as you know, membrane protein is important for maintaining the integrity of the cell membrane, right? Because they provide attachment points for cytoskeleton and so forth. So once you denature the protein, you basically destroy the cell membrane completely. And that's when the temperature peaks. That's when the absorbance peaks, I mean, at 70 degrees. A lot of you put 80, but the peak actually occurred before. Okay, like you just get like minor increase afterwards. You're no longer rising rapidly, right? And so at this point, cell membrane is completely gone. Cell membrane is basically destroyed. So even if you heat it up further, you're not like everything has leaked out already. You heat it up more, you're not going to get more color, right? So what do you expect the absorbance to be? If we go higher temperature, right? No change. Okay. Everything is already leaked out. Has already leaked out. Okay. That's it. I think those are the key things you have to know for the lab when you're doing the test. Does anybody have any question before I move on to lecture 1? Okay. What is a placebo? Give an example of an experiment where a placebo is needed. So placebo is basically a sham treatment, right? Sham treatment has no effects on the dependent variable. Okay. Give an example of an experiment where placebo is needed. Usually when you deal with humans, right? When you involve humans. Involve human. To eliminate, right? We have to minimize bias and psychological effect. Psychological effects. When do we not need a placebo? Animals, right? Animal testing. Remember the example we did in class with the cows, right? Like if you give them more proteins, is there going to be a change in the milk yield? Right? So we don't need a placebo group. And cows don't have bias, right? What is a valid hypothesis? Anybody? Two things that must be true for a hypothesis to be valid. Must be what and what.
Measurable.
Okay, measurable. Anyone else? Okay. RJ say falsifiable, yeah. Falsifiable. And then what's the other one? Not quite measurable.
Testable.
Testable. That's right. You got it. Okay. Practice writing hypothesis. We did quite a bit of that, right? So in the case of the beetroot lab, it would be increasing the temperature is going to increase membrane permeability. Okay. So not so much practice writing because there, like I said, there are no written response question this time. You don't have to type anything. All of them are just multiple choice, right? But you do have to recognize what's a good hypothesis, what's a valid hypothesis, right? Like things that involves like opinion, ethics, those kind of things are like not really hypothesis, right? Okay, look at the test results below for testing the efficacy of the drug for reducing jet lag symptoms. Okay, so over here, what's important to notice is because the placebo and the drug both work equally well, that tells you it's not the drug that's reducing the jet lag symptoms, right? It's just psychological effects, right? So both placebo and the drug reduces symptoms, right? So the drug is not effective, right? Like I could just take a candy and I could achieve the same effect. The drug is useless. It's not doing anything. On the other hand, over here, only the drug is able to reduce the symptoms. Right? So it is effective. The placebo is not supposed to do anything, right? And the result shows exactly just that. Taking the placebo did not do anything. I mean, it's slightly lower than the control, but like it's not a significant reduction, right? So this is not like an exhaustive list of everything that you should know. It's like a good starting point. If you are tight with time, as some of you are probably, experiencing a high volume of assessment this week, right? With chemistry, math, and probably some other subjects, right? So like if you haven't studied bio yet and you don't have a lot of time, then this sheet, it's gonna get you a decent grade, okay? It's gonna get you a B or something. But then like there are other things that you should go through as well, right? So things like other things such as like the Rady experiment and Pasteur experiment, right? Remember those with the flies and the flasks? There's also like blind versus double blind experiment. Blind experiments, like the subjects don't know what they're receiving. In double blind, the person who's administering the test also does not know, right? So this helps minimize bias. So, you know, if I ask you, what are some of the measures that can be taken to minimize bias? Not only do you need a placebo, but you also need to make sure that the experiment is done, you know, in a double blind setting. Primary versus secondary sources. Okay, so like a few extra things that you should be mindful about. Anything from lecture 1 you want me to go through? Anything else?
Sir,
Mm-hmm.
which is which again? Like the reddish one's like the meat, right?
Oh yeah, the radi is the fly and the maggot, right?
Okay, thank you.
Yeah, and then the pasteur is the, you know, the toothless. This one, and then the one with the swan neck, right? So they are both experiments that test the spontaneous generation theory. You know, just go through the poll questions that we did in class, right? What's the dependent variable? What's the independent variable? I told you guys, right? Like you can download all the poll questions in a single file, right? And some of those poll questions will show up on the test. So make sure you go through them yourself, right? Make sure you know the
Excuse me,
answers.
sir. Where can we get the poll question?
It's already posted online. You didn't see it?
No, sir.
Let me see if I can show you guys. Just read the announcement after each class that I post, right? So here, test 1 review has been posted. That's the one we're doing right now. I also put all the poll questions in a PowerPoint file. You just click on it.
Okay, thank you, sir.
Yeah. Now you cannot see the answers here, but once you download it, you can see it. Okay, like in a PowerPoint, once you open it, right, like at the bottom there's some notes. That's where I put the answers. Okay, strongly recommend you download this and go through the questions yourself. There are 48 questions, okay? You go through it all yourself. Okay, it's going to be beneficial. Okay, macromolecules. What holds the atoms together in a water molecule? So if I have H2O here, right, this is a covalent bond, right? Covalent bond. And in this case, in the case of water, we have uneven sharing of electrons. And because of that, part of the water molecule is negative, part of it is positive, right? Overall, the molecule is still neutral. So how would you describe a water molecule? It is neutral. Okay, but polar. Polar means, again, part of it, the oxygen is slightly negative, and then the hydrogen is slightly positive. When you put two water molecules next to each other, the oxygen and the hydrogen will be attracted, right? And this interaction is called hydrogen bond. Hydrogen bond. So later on in other lectures, in other topics, we keep on bringing back hydrogen bonds again, right? Do you guys remember what context we did that in? My morning class was having trouble answering that. What do you guys think? Well, what other molecule has hydrogen bonds in it? Molecules that we've talked about in great detail. Yeah, protein, right? Thank you, Justine. A protein, right? We have hydrogen bond that holds the structure of proteins together. We have hydrogen bonds also in DNA that holds the nucleotides together. Okay, so more on that later. But that's basically, you know, water molecule. Anything that dissolves in water is hydrophilic. Right? Hydrophilic. And then the opposite is hydrophobic, right? So that would be like oil, wax. Right? This would be like sugar, salt. Okay, hydrophilic, hydrophobic. If you supply radioactive nitrogen atoms, something that can be traced and detected, to a cell and let it grow, what molecules will you expect to find the radioactive labels in? This is basically a question that asks you what molecule contains nitrogen. Does anybody know? Of the 4 macromolecules that we learned, which ones contain nitrogen?
Protein.
Protein, yeah, that's right. Protein is made up of CHNO plus a little bit of sulfur. What other molecule?
Nucleic acid.
Nucleic acid, that's right. Nucleic acid, which contains CHNOP, right? So you would expect to find a radioactive label in these molecules. All right, DNA. What is the monomer for DNA? We have the nucleotide. Nucleotide, which is made up of 3 parts. There is the phosphate. There is a sugar, and since we're talking about DNA, it is going to be a deoxyribose and a nitrogen base. Nitrogen base. And in the case of DNA, what are the possible nitrogen bases? Anybody remember? That's right, Jay. ATCG. Very good. There we go. How are the nitrogenous base classified? So how do we group them together, right? We basically have 2 groups. The purines, which have 2 rings. And those include A and G. And we have the pyrimidine. Pyrimidines, which is only 1 ring in their structure. And that would be the T and the C. What is the base pairing rule? Base pair rule. A pairs with T, right? And C pairs with G. And you might remember A and T has two hydrogen bonds. 2 hydrogen bonds holding them together, but C and G, they have 3 hydrogen bonds. So if you have a 2 DNA strands with a lot of C's and G's, it will take more energy to pull them apart because they are tighter. They're wound up together a little bit tighter than the ATs. What force holds the nitrogen base together along the length of DNA? I just told you, right? It's the hydrogen bond. Hydrogen bond. Okay. And they can be disrupted. Can be disrupted by heat. If you heat things up, you will break the hydrogen bond. Right? So think of water. If you heat it up, you break the hydrogen bonds. When you break enough hydrogen bond, then the water molecule escapes from the liquid, goes into the air, becomes steam, right? What is the significance of the base pairing rule? Well, the fact that A pairs with T and C pairs with G's means that the purines always pair with a pyrimidine. Purine always pair with a pyrimidine, and this creates a constant width along the DNA length. Okay? Like if you think about the DNA here, right? It's always the same width. Always the same width. Okay. Purines are 2 rings, pyrimidines is 1 ring. So the width is always 3 rings wide. This is of course twisted up, right? Like I guess it's easier if you pull it apart. Like don't twist it. Okay, same length across. Okay, so in this ladder model, the psi of the ladder is the sugar phosphate. Sugar phosphate backbone. And then the rocks in the middle, that's the base, right? All right, feel free to stop me if I'm going too fast or I'm not making sense here. What is dehydration synthesis? So you might remember if you have monomers and then you have polymers here, you need to connect them together. And the process for connecting together is dehydration synthesis. Okay, mix water. You're dehydrating and you're taking the water out. And then the other way around, that is hydrolysis, right? Hydrolysis. You have to add water molecules to it to break the bond. And it's the covalent bond that's holding these things together. We'll do some practice question later, okay? Hang in there. What is an enzyme? Enzyme is made of protein, right? Protein that catalyze, speeds up, right? Catalyze chemical reaction. In a cell. How does an enzyme work? So it lowers activation energy. Remember, it takes energy to get a reaction going. Okay, you can just start it from nothing, right? So sometimes that requirement is too high. with the help of an enzyme, you lower that energy requirement. Okay, and how does the enzyme work? Well, you've seen me draw an enzyme before. This is an enzyme. And you have these grooves, these pockets called the active site. That's where you would put the ingredients in, right? The substrate. So the substrate has to fit the pockets. Right? Substrate. The shape has to be matched up. And then the enzyme will do the reaction. It might break the bond in the substrate. It might transfer a phosphate from one molecule to another, as we saw in the substrate level phosphorylation, for example. Yeah. That's basically how it works. What factors affect the functionality of an enzyme? Well, enzyme is made of proteins. So anything that changes the shape. A fancy word for shape is conformation. Anything that changes the shape or conformation of a protein is going to affect the function of an enzyme. Okay. Help me out here. What are some factors that affects the shape of a protein?
Heat.
Yep, definitely. So temperature. Anything else?
Substrate concentration.
You are correct, but we did not talk about that one. You probably read that somewhere else, right?
Yes.
Anything else that we talked about in class?
About pH level.
Yes, right. pH, right? Salt concentration, right? Types of solution, right? So on and so forth, right? If you read about other things, great. But these are the main ones that we talked about in class. What does the enzyme lactase do? Well, anything that ends with ASD, it's probably an enzyme. Not always, but like most of the time is an enzyme. So lactase is going to take lactose. Does anybody remember what lactose is made up of? Lactose is a disaccharide. It's made up of two things. What are they?
Glucose and galactose.
Excellent. Glucose plus galactose. Right? But it won't break down on its own. You need an enzyme for that, right? The enzyme is called lactase. That's going to break the bond, the covalent bond that holds them together. Add a water molecule to it. And you get your glucose and your galactose. Okay? Complete the following chart.
I have a question, sir.
Yeah, go ahead.
When the enzyme lactase breaks down the glucose and galactose, the breaking down, that's dehydration synthesis?
When you break things down is hydrolysis, right?
Oh,
Yeah. When you build things is dehydration synthesis.
Okay, thank you.
Okay, yeah. So also along that line, when you break things, that's catabolism, right? When you build things is anabolism, right? We learned that in lecture 4. All right. Carbohydrates. The monomers for carbohydrates, we collectively call them monosaccharide. Single sugar, right? Which include glucose. And like this would be a nice chart to have. Like next to you, right? During the test, if you have trouble memorizing these things. Glucose, galactose, and fructose, right? Biological functions. They are like main source of energy. We learn in lecture 5, right? Like how we break down the glucose. We get the ATP from it. It's main source of energy. We also can store energy, right? Using carbohydrates. And for some organism, they could do structural support. Example? What are the 2 examples for energy storage? What carbohydrates are used for energy storage? Anybody remember?
Can you repeat the question?
Glycogen.
Glucose.
Glycogen is a good one. Yeah.
Fructose.
Yeah, hold on. In muscle and liver. Not glucose, right? Not fructose. Those are simple sugar. We just burn them right away, right? Those are for immediate usage, not for storage. What about for plants? What is the storage form of carbohydrates in plants?
Cellulose.
Cellulose is for support. Chitin is for protection. There's only one left. Which one is that?
Sucrose.
Glucose is for like immediate burning, right? For fuel, right? Like we're not storing that one, right?
Chlorophyll.
Which one is that? Sorry, one more time.
Chlorophyll.
No, chlorophyll is for capturing light to produce food. That's okay. I will just tell you guys. Which is why we're having this review, right? Starch in stem and root, right? Hopefully, when I write that, you're like, oh yeah, I remember, right? Okay. So make sure you have this chart. I know it's a lot of information, right? When's chemistry, anyways? Is chemistry tomorrow for you guys?
Yep.
And then math is the same day as me.
Yes.
Oh
Yep.
boy, and mine is the last one, right? Oh my goodness, that is just very unfortunate. Anything else on Friday? Do you guys have anything on Friday?
No.
No.
No? Okay. Do you want me to move the test to Friday? Is that gonna help?
Yes.
Yeah, I mean it's fine for me. Yeah, okay. I'll do that. Why don't I do that? Okay. So then you guys have like an extra day to study. Okay.
Thank you, sir.
Yeah,
We
you're
like you,
welcome.
sir,
Thank
so
you,
much.
sir.
Thank you, sir.
No
Can we
worries.
love
do it
you.
anytime on Friday?
Yeah, Friday from 6 to you know what, I'll actually extend it. Okay, so like it's available on Thursday. So those of you who want to get it over with, that's fine. But then I will extend it all the way to Friday night. Okay, Friday
Sure.
like midnight. Okay,
Okay,
so
thank
that
you.
way you will have that option. Yeah. Okay, now you like me, right? Yeah. Before it was like so. Anyways,
Either Friday or either tomorrow or Friday.
Friday, right? Friday. Right. So it will be available on Thursday, like we originally planned, but you will have up to the end of Friday to do it. Does that make sense?
Yes.
Okay, great. Structural support. Structural support, we have things like chitin, right? Chitin for in like shells, right? Like lobsters, crabs, right? Insects. And then the other one is cellulose, which is in cell wall, right? Cell wall. Okay, so for protein, I mean that's it, right? These are, by the way, these are all polysaccharide, right? Polysaccharide. Okay, next we have proteins. Right? You should know by now that the protein is made up of 20 amino acids, right? 20 amino acids. And I mean, you don't have to draw anything on the test, but you
Yes,
should.
sir. Do we have to write 20 amino acid, or we should just write amino acid?
I mean, like, you're not gonna be writing anything, right? So it's all gonna be multiple choice.
Okay.
If I ask you how many amino acids make up proteins, right? You know you have to choose 20, right?
Okay, thank you, sir.
So you know we have the amino group here. We have the carboxyl group, and then we have the side chain, right? So what makes one amino acid differ from the other is the side chains. All right. The biological function for proteins, that is going to be, I mean, there are a lot of functions here. Let me list them all out for you. Energy source. When we eat meat, or like if you eat like soy product, if you're vegan, right, or vegetarian, you eat soy product, you eat beans, right? Those are energy source. We need proteins for things like enzymes. Right? We have membrane proteins. Remember membrane proteins in lecture 4? At the beginning of lecture 4, I went through 6 functions with you, right? For membrane proteins. Attachment to cytoskeleton, cell signaling, cell identity, transport, right? So all those things, right? That's membrane proteins there. We can review some of those later. We also need proteins for hormones. Okay, some hormones are protein, other hormones are lipids, right? And we also can have proteins for structures. So energy source is just like, you know, like from meats, right? Meat, beans, soy products. Enzymes such as insulin, it helps you absorb sugar from the blood afterwards. Membrane proteins, I mean, they have various functions, right? 6 functions in total. Let me just quickly show you what I'm talking about. It was lecture 4, and in the study guide, you know, like these ones, right? Attachment to cytoskeleton, cell signaling, enzymatic activity, intercellular junction, cell recognition, transport, right? So all these are functions of membrane proteins. You should probably go through it yourself, but you know, let's say transport, right? Transport. Does anybody remember example of transport that requires membrane protein?
Hemoglobin.
Which one is that? Sorry, I just
I'm
didn't
not
hear you.
sure. I'm not sure. I
Okay.
said hemoglobin.
Oh, hemoglobin. Oh, hemoglobin is for transporting oxygen.
Is it
that-
like active transport?
Yeah, sure. Right. Do you have a specific example of active transport that involves a membrane protein?
Maybe like water dissolving through the cell membrane, like that?
Water
Not
moving through.
dissolving, like passing through
Right.
and
Yeah, that would be osmosis though, right? But good try, good try. Protein pump, right? Sodium potassium pump. We wrote that down. Yeah, there's going to be some kind of integral protein such as carrier protein and channel protein, right? And carrier protein is the Pac-Man, right? The one that looks like this. It grabs onto the molecule and then it spit it out on the other side, right? Channel protein is the one that creates like a tunnel for things to go through. So we'll review those later as well. Hormone. Oh, I'm sorry, I made a mistake. The hormone is the insulin here. I read that wrong. Right? Enzyme would be like lactase, right? That we talk about. Enzyme catalyzes reaction. Hormone, it's like communication between different parts of the body, right? And structure would be like muscle, hair, nails, so on and so forth. All right, there you have it. That's the protein. Lipids. Lipids does not have monomers. Instead, we have different components. So like the glycerol, the fatty acids. Let me spell that out for you. Fatty acid. Right? You can have the phosphate group, the polar head. And the carbon rings, right? Lipids are excellent for energy storage, long-term energy storage. Glycogen is more for short-term, right? Like if you don't eat, if you skip dinner tonight, you're gonna be fine. You're gonna burn your glycogen reserve. Lipid is for like, you know, if you are fasting over a long period of time. We need lipid for like insulation and protection. We also need lipids for like cell membrane, right? Cell membrane structure. Okay. And some lipids are hormones. Let's not forget that. So for energy storage, it's just fat. You know, surrounds the organ, for example. Yeah, BB, go ahead.
I just wanted to ask if you could expand on protection.
Yeah, sure. Like many organs have fat surrounding them. So when it's minus 40 degrees outside, right, the fat basically keeps them warm. So that's the insulation part. Fat is very good at trapping heat. And in terms of protection, like, you know, if I fall, right, the fat sort of acts as bubble wraps around the organ. So it absorbs the shock, right? It's not going to bruise your organs or stuff like that, right? So that's what it means. Cell membrane structure. Does that answer your question?
Yes, thank you.
Okay, so cell membrane structure, we have the phospholipid. We also have the cholesterol, right? And then hormones, the sex hormones that we talked about, estrogen, right? Testosterone. Remember, these are steroids, which made up of carbon rings, right? All right, moving on, nucleic acid. Nucleic acid. We have two types of nucleic acid, DNA and RNA, and the building blocks are nucleotides, right? So for DNA, someone already wrote this down earlier in the chat box. And for RNA, which letter are we switching out? Yeah, exactly. We're switching out the T and it becomes a U. There you go. In terms of biological function, it is genetic material. Passed on to next generation. Okay. Also contains instruction. Oops, can't spell. Sorry. Instruction to make proteins. And I mean, we only have two examples, right? Like DNA, which is in the nucleus most of the time, and RNA. Right? We learned two types of RNA: messenger RNA for making proteins, and rRNA, which makes up the ribosome. All right. How is the shape and structure of membrane stabilized? Remember this thing, right? The pipe cleaner that I was using to explain that. Man, that seemed like a long time ago. But let's go through it together one last time. So for primary, it's basically the amino acid sequence, right? That's the amino acid sequence. And what holds these amino acids together is covalent bond. Right? All the macromolecules essentially are held together by covalent bonds. Now you can fold this thing up, and the secondary structure are the helix and the beta sheets, right? So these are alpha helix. These are beta sheets. And it's stabilized by hydrogen bond. Hydrogen bonds in the peptide backbone. All right. Long time ago, right? Like in lecture 2, I drew you a picture. You gotta go back to the recordings, right? And I say, the yellow ribbon represent the backbone, right? And then with all the side chains coming out, I don't know if that rings a bell. But the backbone can interact with each other by forming hydrogen bonds, and that will create the helix or the beta sheets. And then you have your tertiary structure, which is like the three-dimensional shape, right, of the protein, right? So the protein kind of folds back up on itself and forms like a ball, right? So this is the 3D shape. And it's stabilized by the interactions of side chains. Side chain is the R. Okay. Now I'm running out of space here, but quaternary, which exists for some proteins, not all of them, it's when you have multiple peptides coming together. Right. Let's write that down as well. So for the quaternary structure, it's like, maybe two pink protein comes together, and then we have two blue one comes together, and it forms a complex, right? This is a protein complex. Right? Like hemoglobin, has 4 parts to it. Right? Individually, the protein subunits are not functional on its own. They need to work together. Right? So that's quaternary structure. Not all proteins are going to have it, only some. And the secondary, tertiary, and quaternary, they are sensitive to pH, temperature, ionic concentration, all the stuff that we talked about. But the primary structures are usually unaffected. Like even if you heat up the protein, the primary structure is gonna be intact. I mean, the protein is gone, but then you still have the sequence, right? To break down the sequence, you need to have, enzymes that are designed for breaking down, right? So that you have that in your stomach, right? It will chop up these amino acids. Okay, so that's that. Next, identify the molecules. This here, hopefully, you know, is an amino acid. This one, we have a phosphate, sugar, and nitrogen base, right? So those three things makes up a nucleotide. This is some kind of sugar, like you don't really know which one. So we just say monosaccharide. And here is just a long chain of hydrocarbons. So that's fatty acid. Okay, like you don't have to draw them, but if I show you a picture, you should know what you're looking at. Okay. Now we don't have time to go through all of these, unfortunately. So list the major components of cell membrane. I'm gonna refer you to the note that we took, which was in lecture 4, I believe, maybe. There we go. The fluid mosaic model, right? Remember this picture that we drew? Okay, so like you should familiarize yourself with that. Okay, I'm just gonna copy it here, right? And paste it right here. Okay, so you have the different major components of it. Okay, I mean, no one's gonna ask you to draw things on the test, but you should know, for example, oh, okay, like there are these cholesterol, which helps maintain the membrane fluidity, right? Glycoproteins are for cell identities, right? So on and so forth. Describe the central dogma of biology. That is starting at DNA, which contains all the instruction, right, to make the proteins, but we don't need all of it. We just need to make a copy of it, a segment of it, right? We copy the part that we need, and that is called transcription. That happens in the nucleus. And then the messenger RNA will leave the cell, leave the nucleus and go into the cytoplasm, where the instruction will be used to make the protein. That is called translation. Right? So that's the central dogma, central theme of biology. One of the functions of the liver is to remove toxins from blood. Based on its function, which two organelles would you expect to be abundant in a liver cell? Detoxification. Which two organelles? Do you guys remember?
Peroxisome.
Yeah, sure. Peroxisome. That's good. That's one. Anything else? Peroxisome.
Lysosome.
Yeah, lysosome. And actually, there's one more. I know they only asked for 2, but then I wasn't expecting peroxisome, but that's a good one. There's one more.
Smooth.
Yeah, you got it. Smooth ER, right? Okay, great. Describe the structure of a mitochondrion and state the function of each part. You must be sick of the mitochondria by now, right? You should be. We talked about it so many times and spent so much time going into all the details in lecture 5. But in case you want another shot at it, this is the outer membrane. This is the inner membrane, which forms the cristae, right? And that is where you will find the ETC, remember? The more foldings you have, the more surface area, the more ETC you can pack into it. This part is the matrix. and this is the intermembrane space. Now, the general idea is we take H plus and we pump it, right? We pump it into the intermembrane space, right? We pump all the H plus in here. Okay, and when they come back in through a special turbine, we make ATP, right? That's the oxidative phosphorylation. We create a damp, we let the water comes back, we let the H plus come back in through the turbine, and we generate ATP. All right. Let's take a quick break, 5 minutes, and then we'll do some practice questions and the rest of the study guide afterwards, okay? See you in a bit. All right, time is of the essence. Let's keep going. Describe the endomembrane system. So again, in lecture 3, we took a note here, right? We took a note here that shows you the endomembrane system, essentially. Okay, so I'm going to go through this picture with you. You already have this. We drew it together. Right, so the endomembrane system includes the rough ER, the smooth ER, the Golgi body, as well as the lysosome. Okay, so the idea is this: the rough ER, working together with the ribosome, basically mix a protein, and then you will package it into a vesicle. The vesicle will travel to the Golgi body, where the content of the vesicle will be sorted, will be modified, and repackaged into a new vesicle. This vesicle can go to various places. It can leave the cell and secrete the protein. We now know that is an example of exocytosis. It can be stored in the cytoplasm for immediate use or for later use. It can become part of the cell membrane, right? Membrane proteins, lots of functions. Cell recognition, transport, enzymatic activity. And it can also become the lysosome, right? The lysosome is responsible for breaking down macromolecules. It is responsible for digesting old and worn out organelles, and it might even trigger apoptosis if you need to kill the cell because it's too old. So that's the endomembrane system. Now, you guys all showed up here, right? And you didn't show up for nothing. So as a way to show my appreciation, I'm gonna give you some questions that might show up on your test. Why don't you take a look at that? I think I'm gonna skip the poll today. All right, you guys are not too enthusiastic about polling for some reason. Right? So if you know the answer, you can type it in the chat box. If not, that's okay. I'll take it up in a bit. Anybody? All right. I have an answer from RJ, which is C. Anyone else? B? Okay, well, let's see. Once a solid material is phagocytized, that means phagocytosis, right? Has been eaten and taken into a vacuum. Which of the following statement best describe what happens? Well, perhaps I should show you the slide that corresponds to this. I think that is lecture 3 Do I have a lecture 3 here? There we go. Right here. Right, lysosome. There is a food that has been ingested, food vacuole, and then the lysosome will come and break it down, right? After all, that's one of the functions of lysosome, to break down food particles. And that is the answer, C. Okay. A lysosome combines with vacuole and digests the enclosed solid material. Ribosome does not have anything to do with vacuole, okay? Ribosome just makes protein regardless, you know, as long as there is amino acid available and the mRNA. Okay, so that is that.
Sir,
Mhm.
I have a question. Is a vacuole and a vesicle the same thing?
Yeah, I mean, there's subtle difference, but for us, it's the same, okay? Vacuole, vesicle. Usually
thank you.
vacuole is referring to things that are coming in from outside. Vesicle usually are like packaged in-house in the cell.
Oh, I see.
Yeah, but
Thank you.
it's interchangeable. All right. Obviously, there are still like a lot of organelles, right, that we don't have time to cover. Make sure you go through them. I don't think I put any questions about cytoskeleton on. I am 90% sure I didn't. So like maybe you don't need to go through that. But definitely know like chloroplasts, right? Centrioles and all the other ones that we didn't talk about. All right, things that are specific for plant cells and animal cells, go through those as well. Okay, membrane transport, right? Membrane transport. Again, if you are gonna have some notes next to you, and you are encouraged to do that for the test, then this is definitely one of those that you want to have. This table, right? This chart, right? Okay, so like this is we spent like almost an hour writing this. It tells you about the passive transport, right? What molecules are transported using that method, right? So like if something is charged, it's gonna use channel protein. If something it's bulky like glucose, then we're gonna use carrier protein, right? These I can ask you a bunch of questions from this chart, right? For example, select all of the active transport method from the following list, right? And then you're like, oh, active transport, let's go to here and then just check off all these things, right? Okay. Like ideally, you should have memorized all these, right? But I'm not naive. Like I know some of you just gonna have the notes with you, which is totally fine. Okay, knowing where to find information is useful, right? Like that's what all of us do anyways. When in real life, when we don't know something, we look it up, right? Okay. So I can ask you, like, which of the following is used for transporting charged particles, and you have to know, oh, that is channel protein. Do you understand that? Okay. So what was the question anyway? I went on a tangent. Explain what passive transport is and provide examples of passive transport. Well, passive transport is from, let's see what the note says. Passive transport is from high concentration to low concentration. Down concentration gradient, right? No ATP requirements. You guys have this chart, right? You remember doing this? I hope you do. I'd be heartbroken if you don't. Describe the difference between various type of bulk transport. Let's take a look at the bulk transport. Okay, so bulk is like large amount of stuff that needs to move in and out, and it's an example of active transport. If you are moving things out, then it's called exocytosis. If you're bringing things in, then it's endocytosis. Remember that vesicle that was butting off from the Golgi body, right? One of its destinations is to get out of the cell. To get out of the cell, we do exocytosis. To bring things in, depending on what you are bringing in and depending on how you are bringing it in, we have different forms of endocytosis. If you're bringing it mostly solid, then it's phagocytosis. If you're bringing it mostly liquid, then it's pinocytosis. And then if you are choosing specifically what to bring in, then that's receptor-mediated endocytosis, right? This has more selectivity to it, right? It's more picky about what's bringing in. Now, even though this is called cell drinking, but you're not like exclusively bringing in liquid. Like there might be something that is in the liquid and you accidentally bring them in as well. I don't know if this analogy helps. Like think of like it's like really dirty water here, okay? Like in the wild, you're really thirsty, okay? You're lost in the woods or something, and then there's like a puddle of water. You have no choice. You have to drink it, okay? But you have a straw with you. So you stick the straw in and you suck up the water, okay? Like you want to get the water only, but because it's so murky, right? And there's so many things floating around, you might accidentally ingested other things as well, right? And that's what pinocytosis is. The cell wants to bring in liquid, but there's so many things that are dissolved in the liquid. You might inadvertently bring in some other stuff as well. All right. Another freebie. Now, this is one of those questions where you look at it and are like, oh, this is so many words, right? And it sounds weird, right? But in the end, it's not quite difficult. You are just comparing pinocytosis and receptor-mediated endocytosis. So let's see. Pinocytosis can concentrate substance from extracellular fluid, but receptor endocytosis cannot. Well, in both cases, you're bringing things in, right? You're selectively bringing certain things in. So you're choosing a particular thing to bring into the cell. And anytime you choose something to put it aside, you're essentially concentrating it. So that's not the answer. Pinocytosis is non-selective in molecules it brings into the cell, whereas receptor-mediated endocytosis offer more selectivity. That's exactly what I described to you, right? Just before. Let's see why the other ones are wrong. Pinocytosis requires cellular energy, but receptor-mediated endocytosis does not. They both require energy, right? They both require energy because they're both examples of active transport. Pinocytosis increased surface area of plasma membrane, whereas receptor-mediated endocytosis decrease plasma surface area. Well, in both cases, you are bringing things in. It doesn't matter how you're bringing it in. Anytime you bring things in, you lose a chunk of cell membrane, right? Like this part here, this was part of the cell membrane, but you pinch it up, you brought it in. So now your cell membrane has less area, right? So this one also decreases, is not correct. Pinocytosis brings in only water into the cell. So like I said, it's like drinking from the murky water, right? You're gonna bring in other stuff as well. It's not always just water. Okay, like I don't know if you guys know what bubble tea is, but if you ever drink that, like you try to drink the liquid, but you sucked up the tapioca as well, right? It's a byproduct of what you do. Okay, so that is that. The cell of a plant grows in very cold environment. We did that one already before, right? Oh, I'm shifting the markings. Let's not do that. For cold environment, if you remember, we needed to make it less solid. So you want to have more cholesterol and unsaturated fatty acids. Okay, that's it. Less saturated, of course. How are active transport similar to facilitated diffusion? Both require membrane proteins. Okay. In active transport, we have the sodium potassium pump that needs proteins. And in facilitated diffusion, we need carrier protein channel proteins. The obvious difference is that one is active, which requires ATP, and then facilitated does not. Right? And that's pretty much it for number 4. Of course, there are other things that we did not review here, which are important. Definitely go through the hypotonic, hypertonic stuff. Here's a question to help you jog your memory a little bit. Right? Hypertonic, right? Hypertonic. So hypertonic is the one that has more dots outside. Right? So it'll be losing water. Okay, so let's see. The cell will show no change due to diffusion of both solute and solvent. That's not true. The only time there's no change would be isotonic. The cells will lose water and shrink, right? That's correct. Right? Shrink is also plasmolysis. Okay? Just some terms that you may want to be aware of. The cell will swell and ultimately burst. No, that's for a hypotonic. The cell will shrink at first, but later reach equilibrium with surrounding solution and return to original condition. Nope. Once they change, they change. Okay. Think of a raisin. The raisin is shriveled up, right? If you put the raisin in the water, it will go back into the grape form, and it will stay that way. Right? It's like if you leave it in water, it's not going to shrink back into the raisin, right? So similar situation here, but opposite. Right? Once it shrivel up, it will stay shrivel up unless you change the environment, right? Okay, so make sure you go through the other cases. Metabolism, we're almost there. What is an organic molecule? Basically anything with C, right? Anything with carbon, but not CO2, CO, or CN. Right? The people who came up with this decided, you know, let's confuse everyone. Let's put these on the exclusion list. List the products of glycolysis. Again, one of the important charts and tables to memorize or have available to yourself is this one. Okay, this one summarizes all the key products. Okay, so I'm going to put them right here. I know the question only asks for glycolysis, but really, you want the rest of them, right? Oh boy, what's going on here? Okay, so glycolysis involves the splitting of glucose into 2-pyruvate, and along the way, you're able to make some ATP through substrate level phosphorylation, and you pick up some electrons, right? In cellular respiration, glucose is oxidized while oxygen is reduced. Explain what this means. This is the thing that I've repeated to you multiple times. Glucose, we're gonna release electron. right? These electrons will be picked up by NAD and FAD, and then these will carry the electron to the electron transport chain. Right? The electron transport chain is a series of proteins. Right? The electron hop off onto the first one, and then it get passed down, and then ultimately at the end you have the oxygen. Right? So glucose is oxidized because it's losing electron. Oxygen is reduced because it's picking up that electron. But it's important to remember, it is not a direct transfer. Okay, the electron has to go through a bunch of intermediates, right? The energy carrier first, and then the ETC, and ultimately, the oxygen is the final electron acceptor, right? If you were here last class, I was telling you, right? Like it's like the parent that pulls the kid off the slide at the bottom, right? What is the role of NADH and FADH2? I already told you. They transport the electron to the ETC. What is the significance of coenzyme A? In pyruvate oxidation, 3 things happens, right? Let me write that down for you again. We're gonna remove carbon dioxide. We're gonna carry some electrons. And I kept on saying, by this point, the molecule is out of energy, right? It doesn't really want to react anymore. It has been through a lot. So to recharge the molecule, right, the coenzyme A is going to be added to it, and it becomes acetyl-CoA. So the purpose is to re-energize the molecule so that it will react further. Okay. There will be calculation questions like these on the test, but in like multiple choice format. So please tell me what the answer is. Plug it in to your calculator. $188 from RJ. Maitri also. Is it Maitri? I'm sorry, 188. Yeah. Same 188. Great. This one times 3 because it's NADH. This one times 2. 3 times 1, 3 times 50 is 150. 2 times 19 is 38. Add them up, 188. Okay, I want everybody to get this question right on the test. I mean, obviously it's not going to be 15 and 19, but then, you know, you will be able to do it. The inner, finally understand this, okay, great. The inner mitochondrial membrane area is about 5 times the area of the outer membrane, right? Because it folds up into the cristae, right? And the purpose of this is to maximize surface area to allow more ETC to be embedded in the cristae. The more ETC you can pack in the cristae, the more electrons can be accepted from the carriers, and then the more H+ you can pump from the matrix to the inner membrane space, and then the more ATP you can generate. Which-
What is the ETC again?
The ETCs is the electron transport chain. Okay, so from last class we had right here, right? Like this is the crystal. These purple circles, those are the electron transport chain. It will pick up the electron from the carrier and use the energy from the electrons to pump H plus from the matrix into the intermembrane space. Okay, so once you have this gradient, they could come back in through the turbine to create ATP. Okay, so the ETC is just a bunch of proteins, right? Check the notes here. ETC, a group of membrane proteins found in the crystal. Which step in cellular respiration generate the greatest number of ATP? The ETC. Okay. 32 ATPs are created through oxidative phosphorylation. Let me just scroll up a little bit to show you, right? Oh, 32 ATPs, right? These are from oxidative phosphorylation as opposed to substrate level phosphorylation, right? So in oxidative phosphorylation, we create a gradient and then they come back in through the turbine. 2 steps to it, right? So describe what happens as H plus is pumped from the matrix into the intermembrane space. Okay, so a concentration gradient is created. It's like damping the water, right? So here, lots of H plus in the intermembrane space. They really want to come back in, but they cannot because the membrane is impermeable to charged particles, right? So that's why we need the doorknob, the ATPase, the turbine, which allows the H plus to come back in. As they come back in, the energy is used to add P to ADP, and that creates ATP. Okay. Finally, the purpose of the final electron acceptor, I keep on repeating this last time, right, in class, right? O2 removes electron from ETC to allow continuous electron flow, right? Remember my analogy. If the kid at the bottom of the slide not go away, then the next kid cannot slide down. If the electron is not removed from the ETC, the next electron cannot slide down. If the electrons are not flowing down the ETC, you're not pumping the H plus. Without the H plus, you cannot create ATP, right? That's why we need to have the oxygen. Like I said, I try to make the question very straightforward for the last lecture because I know it's like very crazy. Hopefully it will help. Okay, I'll give you 2 more free questions. When I say free question, I mean like they could appear on your test. Okay, there's not guarantee, but there is a chance because it's all randomized, right? So I don't know like if you're gonna get it, but let's try this one. What do you think, huh? Very good. Anyone else? And again, if you are confused, let me redirect you to the notes. That's why it's important to reorganize the notes before you take the test. Here it says, right, I wrote it in pink. Hopefully you did too. To phosphorylate a molecule means to add a phosphate to it. So that's the answer, right? We also learned a bunch of poison. See if you remember this one. This is DMP. That's the one from the factory, right? Like the guy accidentally ingested some and then he had like rapid weight loss. Remember that story? But it turns out to be poisonous. Why? Yeah, this one is tricky. Like I mean, like it's okay if you don't remember. Like it's one of those like minor details, right? So these are all the poison. Let's look at the picture. DMP. Right? Look at the picture. Right? And then look at the question again. Okay. What do you think? Let me put the question right next to it. Do you change your mind after looking at this picture, even without me explaining? No? Well, this suggests that the H plus is leaking back in, right? Right, so that is the right answer. A, making the inner mitochondrial membrane porous, leaky. That's how the H plus comes right back in. Okay? It's not inhibiting the movement of H plus. It's actually providing like a shortcut so that the H plus is not going through the ATPase. And if it's not going through the ATPase, you're not making ATP, right? And then your body thinks you're starving. That's why I switched to fat metabolism. That's why you lose so much weight all of a sudden. Do you understand? Okay. Disabling proteins in ETC, that would be like rotenin, right? CN and carbon monoxide. There's actually a slide, 2 slides, right? Sorry, a couple of slides here, right? It lists all the poisons and how they block, how they disrupt the ETC. Okay,
So what's the correct answer?
that's the correct answer. After all that, look here, look at the picture. What is it doing to the hydrogen? Right, it allows them to leak back in, right? Makes the inner mitochondrial membrane more porous, leaky, right?
Okay, so
All right, that's all for the review. Any last minute question before we say goodbye? I won't be seeing you until after the reading week, right? Next week is like you deserve a break. You did so many evaluations this week, no test, nothing. And when we come back, it's a new beginning. We'll start the unit 2. Okay. After this test, none of the stuff will ever be tested again, right? For my course. Any last minute question? Well, if you do, you know where to find me. Just shoot me an email. And best of luck with Chem, Math, and maybe English and computers and other things. And of course, on mine as well. Okay, take care, guys. Bye-bye.
Thank you.
sir.
Yeah.